Hello and welcome to this webinar titled Using In Vitro Dissolution to Support Post-Approval Changes, Global Regulatory Expectations, hosted by Biopharma Asia Magazine and presented by Xin Amy Bu, Principal Scientist at Bristol-Myers Squibb. My name is Stephen Edwards and I'll be your moderator. Now please allow me to introduce our presenter. Dr. Bu is an Associate Director of Global Regulatory Science Department at Bristol-Myers Squibb Company. She has more than 20 years of experience in pharmaceutical development and regulatory affairs. During her tenure in pharmaceutical development, Dr. Bu led several IND development projects by leading integrated CMC teams to advance development candidates from preclinical to phase 2. She also led numerous analytical teams to support projects from preclinical to commercialization, including 2 successfully marked products, Rayataz and Daclinza. She has extensive experiences in project management, analytical method development and validation, and technology transfer. She is an expert in using dissolution methods to demonstrate bioequivalence and applying waiver for solid dosage forms. In her current role as Regulatory Affairs Scientist, Dr. Bu is responsible for setting regulatory CMC strategy and managing investigational and marketing applications, as well as post-approval submissions. Dr. Bu holds a BS degree from Peking University and a PhD in analytical chemistry from the University of Washington. I will now be handing over to our presenter. Welcome, Dr. Bu.
Hello, everyone. Good morning, good afternoon, and good evening. Thanks for Stephen for the introduction, and also I would like to thank Biopharma Asia for giving me this opportunity to share my experience in using dissolution to support post-approval changes. So in today's webinar, I'll focus on the understanding, let me advance the slides. So I'll focus on the understanding of the role of dissolution tests in applying for Biowaiver or demonstrating bioequivalence for post-approval changes. So I'll start with introducing some basic concepts in terms of Biowaiver and bioequivalency. Excuse me. And then I'll followed by some often referred to guidances, especially from major countries like the US, EU, Canada, Australia, and also some Asian countries because their regulation tend to be significant different in terms of requirement, regulations, and data requirement from the major market. Then I'll go over some details on how to apply dissolution to Biowaiver or support bioequivalence using some examples. So I guess before we get started into the details on the concepts and examples, the question you may ask why we want to make changes after the initial application get approved. As you all know that to develop a new drug and put it on market, it usually takes 5 to 10 years, right? Lots of the new medicines, they are for unmet medical need. So the earlier the medicine can be introduced to the market, the more lives we can save. Therefore, during the initial submission, it may only contain a limited choice of formulation, strengths, and presentation, and also because of the short development time, the manufacturing process may not be optimized. Post-approval also include lots of site changes because that usually is a business need. Company may move to a different manufacturing site within the company or decide to contract to a contract manufacturer. So after you make a change, the submission would be focusing on to demonstrate the equivalence between the post-change product to the prior change, before change product. That is so-called established bioequivalence. So now let's go through some basic concepts of bioequivalence and the waiver. So the bioequivalence is defined as absence of significant difference in the rate and the extent to which the active ingredient or active moiety in the pharmaceutical equivalents of pharmaceutical alternatives becomes available at the site of drug action when administered at the same molar dose under similar conditions in a appropriately designed study. This actually includes both post-approval changes, also the for generic products. So it's a generic definition. So as we mentioned, for post-approval change, all broader terms for in the generic company to demonstrate their drug is equivalent to the innovator. It is important to establish equivalence. So it's mostly the demonstration is mostly done by conducting in-vivo bioequivalence study. It's a human study. But in some situations, which I'll explain in more details, a waiver can be applied. Things going slow. Okay, so what is a in-vivo BE study? It is, as I mentioned, establish equivalency between the two products, the post-approved post-change product to the pre-change product. In a BE study, usually a sponsor company compares the systematic exposure profile of a test drug product to that of a reference product. For the two oral administered drug products to be equivalent, the active drug ingredients or the moiety in the test products must exhibit the similar rate and extent of the absorption as the reference product, as per a statistical criteria for bioavailability. So there are different case situations you can apply waiver to waive this in vivo study. One would be based on dosage form. Another is based on BCS classification. And the third is based on dose proportionality, which means that you have same or similar dose form and with different strengths. So my talk will be more focused on the last one. So why Biowaiver, right? So it's obviously for many reasons. One of them is the financial burden. The BE study is costly because it's a human study, and also it takes time and significant resources from design the BE study, recruit volunteers or patients, execute study, perform the blood test, and then do the statistical analysis. So if this requirement can be waived, is usually by the comparative dissolution, which we'll go into details, that the study would only take a couple months to 2 months in the laboratory, so it saves both money, resources, and time. Now let's go through some of the guidances. This is by all means a all-inclusive guidance. I'm basically focusing on the bioequivalence and biowaiver related guidance. This is this page is focusing on the U.S. guidances since we're a U.S.-based company. The first one is a basic general consideration that lay out the concepts in bioavailability and bioequivalence, and then there's a specific guidance that's on waiver of the in-vivo study that's purely based on the BCS classification. The third one is most often referred to so-called SUPIKE guidance. That is for post-approval changes. It lays out the type of changes, the supporting data required and the type of submission needed. Since we're talking about using dissolution to do Biowaiver or post-approval changes, so I also listed two of the commonly used or referred to dissolution guidance. There's an equivalent guidance in USP for the Biowaiver, bioequivalence. So this page lists other regulations. There's ICH, WHO, those are still in draft form. And EU, Australia, there's also Canadian guidance, which I didn't list here. I specifically list a couple of Asian country guidance. As I mentioned earlier, they tend to be quite different, especially Japan is a very detailed guidance. We'll touch on a little bit later. So now let's get into the dissolution part of it, right? The role of dissolution in establishing bioequivalence. So in terms of a oral administered drug, right, it goes through three major steps to be absorbed by the body and to be efficacious. So it is, first of all, it has to be release the active drug substance. So, for example, for tablet form, it's often referred to disintegration. And then it has to be dissolved or solubilized in a physiological conditions. And then the last step will be permeation across the GI tract. So the first critical step can be simulated by a dissolution test. That's why the dissolution can be used to waive bioequivalence study requirements. So I'm not sure in terms of the audience how many are very familiar with the dissolution test. So I'm just going to quickly go through some basic concepts. So in pharmaceutical industry, a dissolution is considered a performance test. It's defined as the amount of drug substance that goes into a solution per unit time under standardized conditions at a set temperature and a solvent composition. So since it's trying to mimic a in-vivo process of disintegration and dissolution, right? So it is usually performed at the body temperature, and the commonly used apparatus is 1 and 2. Dissolution test is actually usually used to guide formulation development, and it is a almost must test for oral drug products like capsules and tablets. But in our case here, we're focusing on using the applying dissolution for post-approval changes. So here are the schematic diagram of the USPS apparatus 1 and 2, basket and paddle. On your right, it shows two dissolution profile. The blue trace is so-called fast profile. It is defined as greater or equals to 85% dissolved within 15 minutes. And the red trace is more or less quote-unquote normal profile, which I'll explain a little bit more later. So before we go into the using the applying dissolution for Biowaiver and post-approval change, let's say what are the changes that the company will potentially make. These are clearly laid out in SUPAC guidances, and also EU and other major countries have equivalent guidance. So it could be change in composition and components, which means you change minor changes in the excipient and in the ratio of excipient to active. Additional strengths that you may have only 1 or 2 strengths marketed as initial application, then later on, depends on the clinical need, there may be added strengths. size changes actually happens quite often because now the companies all becomes global. It's purely driven by business need. With the site change, it often comes with other changes because of the new site may have slightly different equipment. Therefore, the manufacture process may have to be adjusted slightly, and also depends the size of the equipment, the batch size may change also. So as you can see, some of the changes require BE study, like the component and the composition change usually considered as a major change. That's almost always require BE study. And some others may not require BE study, but you still have to provide dissolution data to demonstrate equivalence. So for those that require BE study, as I mentioned, there is opportunity to apply for a waiver. Most commonly used Biowaiver is by applying for the additional strengths post-approval. And most of the major countries have relatively similar guidance on what type of submission and data requirement. So in terms of, there's other requirement in terms of demonstrating equivalencies, such as the quality aspect, which means potency and impurities, appearance, right? But dissolution is a performance test. Basically, you're trying to demonstrate in vivo that after you make the change, it still acts exactly or very similar to before change. So the data requirements for dissolutions really depends on what type of change you're going to make and the country. So, for example, as mentioned, a manufacturer change, it doesn't require a BE study, but it does require comparative dissolution, which is generally only required in one media, which is the QC media that you filed at your initial application. If it's an older product, for generics, especially that if there's a compendia monograph, then it's the monograph method. But other changes may have more requirements, such as like the apply Biowaiver for additional strengths that most of countries require you to do dissolution in multimedia. The three standard USB dissolution condition will be pH 1.2, 4.5, and 6.8, in addition to your QC method. But again, Asian countries, especially Japan, has a lot more requirement, not only more media, more paddle or basket speed, and also longer dissolution time. So once you have the dissolution profile, how do you judge whether they are equivalent or not? There are two ways, depends on the profile. If it's a fast dissolution profile, which means, again, 85% or greater dissolved in 15 minutes, then visual comparison can be applied, which is definitely not preferred. So the other way is by statistical analysis. It's called similarity factor F2 calculation. In that case, that the dissolution profile is less than 85% dissolved. And I'll show more requirement details on the F2 calculation criteria. In order to qualify to perform the statistical analysis by F2 calculation, your dissolution profile has to be a so-called gradual profile, which means you have to have at least 2 time points less than 85%. When you do the calculation, the minimum time point is 3 time points. The more is considered better because then statistically it's more meaningful. If you're familiar with dissolution, you know one set of dissolution use 6 dosage units. So in order to average out the individual dosage unit variation, then this calculation requires 12 dose unit dissolution, which means 2 sets of dissolution, and you take the average of pre-change and post-change profile and do the statistical analysis. So this is just the equation. I'm definitely not a statistician, so I'm not going into it. So the bottom line is you perform the calculation between the two profiles, and then you look at the value. If the value is greater or equals to 50, which is statistically showing they are similar. If they're less, then it is different. So here I just want to kind of illustrate the difference between a specification and the F2 calculation criteria. So as you know, like for any drug product to be released to the market, it has to go through a series test and has to pass specification. For solid oral dose, there's always almost always have a dissolution test unless it's a BCS class 1 product. This disintegration test could be substituted. So for an immediate release, usually the spec is set at 30 minutes. In this example, the spec is 85% dissolved at 30 minutes. So if you look at the data table on the right, all 4 batches would pass specification, which means if they were commercial product, they can be released market. But if you look at the very last column per F2 calculation, the 2 test product 1 and 2 are statistically similar to the reference product. But the third product, although it meets specification, it is considered different from the reference product. So if you were making a post-approval change and you have the reference product as the profile on row 1, and then test number 3 test product, then you will not be able to get this change approved. Therefore, if you know you're going to have post-approval changes coming, when you develop the dissolution method, it is preferred to have a dissolution profile, which means that you should have at least 2 time points less than 85% dissolved. This will be more easy to compare to find if the 2 products are similar or different by the dissolution testing. However, you don't want your dissolution method too discriminatory. What means too discriminatory is that when you manufacture a drug product, there's always going to be slight difference between batch to batch. It's, you have different batch of API coming in, different batch of the excipient, the environment, the equipment may be slightly different, right? So if you're picking up difference that in vivally they don't show difference, then your method is overly discriminating. That's to your company's disadvantage. Then you would not be easily get your product post-approval change approved. So let's go through some examples. As I said, there are 2 often made changes post-approval. One is additional strengths, the other is side change. So that's why I'm providing 2 examples, one of each. So for a additional strength, Biowaiver is possible, especially for lower strength. That some of the regulation actually specifies that Biowaiver is only applies for lower strengths. That really require a multi-media dissolution test. So it's basically 4, the 3 compendial pH and also your QC method. So here we have 2 dosage strengths. The higher strength was approved, and then the lower strength is a common granulation from the higher strength, and is half of the higher dose. So we applied for a Biowaiver. I'll show some data next slides. Just want to mention Biowaiver app, so-called Biowaiver app, is that you have a lower strength approved, and then you want to use Biowaiver instead of A/B study to get the higher strength approved. Guidance has some requirement that you have to meet, and usually it is not well recognized that the Biowaiver app is possible, but our company did have one success that we had a upper strength. We applied Biowaiver in U.S. and is approved by the FDA. However, when we applied for other countries, especially Asian countries, it wasn't accepted, so we have to perform A/B studies. So here's the data, right? So this is a good example to demonstrate here because it is a basic drug. It has a pH-dependent solubility profile. So in this 4pH comparison, we do both have the fast dissolution profile, so which is compared visually, and also has a gradual profile. Some you can see because of the pH dependency, it has very limited amount dissolved within the time frame of dissolution test. However, by F2 calculation for the other conditions, they all pass the 50 criteria, which means they are equivalent. With this data, we were successful to get approval around the world. So what about the higher strengths, right? As I mentioned, you may have to do a B study. So in one situation, we actually did for a different product, we actually did a B study, and it statistically demonstrated bioequivalence. However, there was an outlier in this study. In one of the volunteers, they could not really pinpoint whether the volunteer actually missed the dose, but actually the reference and test product, the absorption profile looks very different. So here we added the dissolution comparison data that to strengthen our justification that the higher strength is equivalent to the lower strength, and that was also accepted by the agency. So the second example is the other most encountered post-approval changes, which is site change. So here you can see that the regulation around even the major countries are slightly different. For example, for a straight site change, which means you don't have a process change, the equipments are all equivalent, and the batch size are within 10-fold. The EU has no dissolution, comparative dissolution requirement. However, US and Australia do have a requirement, which is only in the QC media, but even that, Australia has more requirement that they require a comparison of 3 pre-changes batches to the 1 post-change, while US only require 1 batch each. Taiwan, on the other hand, requires 3 pH media. Interesting, you don't necessarily have to compare it in the QC method. You just have to do the 3 compendial pH dissolution, which is 1.2, 4.5, and 6.8. So on the left is a real example of our product. The pre-change product shows a fast profile, which means at 15 minutes is already greater than 85% dissolved. The post-change, however, is less than 85% at 15 minutes. So as I mentioned before, if both profile are fast profile, visual comparison is acceptable. In this case, it's kind of a gray area because the pre-change profile does not qualify for F2 calculation comparison, but the post profile is not a fast profile. So in this situation, we supplied additional data, which is a prior B study data that for the post-approval change, the site change obviously is the same formulation, same product, we're just moving aside, right? Here we have prior data is a phase 2 and phase 2 formulation comparison. These two formulations are similar in the type of excipients, however, the drug load is significantly different. As you can see that they demonstrate a significant different profile. When we do the F2 comparison, it is less than 50, it's a 39, I believe. So we use this data to show that even in vitro dissolution, showing the two products are different, the B study demonstrates the two products are acting exactly the same in vivo. So using this data to support the dissolution profile difference in the new site and the current site, that should not be different in vivo, which means that in vivo performance should be the same. And with this justification, it was also accepted by the countries this product was submitted. So the third one is not exactly an example. It's just I want to re-emphasize because we did actually have quite some painful experience with Japan. They are not only the dissolution execution of the dissolution test is more complicated and more stringent requirement. For example, the F2 calculation, they do use the same statistical equation to do the calculation. However, it's not a strict comparison whether it's greater than 50 or less than 50. It depends on the profile you have, that it may be 55 or 60, and if you look into the Japan guidance, there's pages, several pages, just to details how to do this comparison. Not only that, there are some, but when we generally consider very minor changes in composition, for example, we have a product. The phase 3 is a pinkish tablet. It's just the coating of the tablet. And when commercial, due to the commercial consideration, the color changed slightly. So it's only a couple of pigments in the coating material changed, but in Japan, that consider a change that warrant a comparative dissolution test. Another product we had that the percent of lubricant, as you know, usually the lubricant is 1% or less in the total composition of a solid dosage form. And the change of the percent lubricant is considered very minor change in other countries, but in Japan it also require a comparative dissolution test. So that's all the example I have. So in summary, a dissolution is a critical performance test for oral solid dosage forms. It plays an important role in post-approval applications. When the dissolution can be used to apply for Biowaiver, it will significantly alleviate financial burdens, reduce time and resources need. Relevant regulations and guidance in terms of dissolution test requirements and the similarity calculations are similar in the major market, but different, especially in Asian countries. The dissolution requirement for the Asian countries tend to be more requirements, stringent criteria, and require more often than the other countries. So that's all I have for today. Thank you very much for your attention.
Thank you, Dr. Bu. Now we will begin the question and answer session of the webinar. But once again, I would like to remind the audience that you can still send your questions in via the questions tab located directly below your webinar screen. And now our first question. What are the appropriate dissolution time points to use for dissolution comparisons via similarity factors calculation?
Okay. if you remember the profile I showed in the presentation, right? So a graduate profile means that you have 2 time points less than 85%. Since the major regulations such as US and EU, they specify 15 minutes as a critical time point. So usually you need to have at least 1 time point before the 15 minutes. Usually we said at 10 minutes. The reason is that it takes time for the solid dosage form to disintegrate. Usually it's somewhere around 2-3 minutes for the capsule shell to dissolve, and for tablet it may be up to 6 minutes for the tablet to disintegrate. Therefore, early time points such as 5 minutes is not really recommended because you have too high a variation. But in order to have 2 time points less than 85%, you should have at least 10 minutes and 15 minutes. Depends on your situation, you could potentially add 20 minutes. The general profile would be 10, 15, 30, 45, and 60 minutes for a immediate release formulation.
Thank you. Our next question is, could you please provide examples of BCS classification and dosage form-based biowaiver?
Okay. So I hope everybody is familiar with the BCS classification. So the drug, the active drug substance usually classified 4 different categories. Class 1 is high solubility, high permeability. Class 2 is low solubility, high permeability. 3 is high solubility, low permeability, and the last one is low, basically low solubility, low permeability. So since the dissolution is a simulation of the first 2 steps, disintegration and dissolution, right? So for class 1 and 3, which are high solubility drug, the requirement of the in vivo study can be waived. That's when the second regulation I shown in the US regulation pages. That is specifically for the BCS classification based. For the dosage form, today we only talk about oral dosage form. The others, you could have injectables, oral solution, oral suspension, patches, and creams, right? So usually the solution, whether it's is an injectable or is an oral solution, those, because it skips the disintegration and dissolution step, those usually you can apply for waiver of BE requirement. Any other questions?
I think we have one more question, which is, what is needed to obtain a biowaiver for a BSC class 2 weak acid, low solubility, high permeability for FDA?
Okay, that's just depends on what change, like I mentioned, right? So if it's actually class 2 is most common drug, at least for our company, we encounter most class 2 compounds. So it's going back to what formulation, assuming it's an oral dosage form, and if you're doing a strength, like basically if you're getting a new strength, you want to get a new strength approved, that is a lower strength, the requirement has to be that they are very similar in formulation. Most of the times they are from the common granulation if your formulation allow you to have a smaller pill. And also I believe the other requirement is that it should have linear kinetics so that there's a dose proportionality. With those satisfied, you can apply for Biowaiver, which is by a comparative dissolution in 4 pH, 1.2 to 4.5, 6.8, and your QC method. And we have multiple cases that it is accepted around the world. So the other change is really depends on, right? So if it's just a side change, usually, as I said, that it depends on which country. For U.S., it is just a straight profile comparison with your QC method. For other changes, U.S. allow you batch size change up to 10-fold, which means your LTSS batch or your initial commercial batch size within 10-fold, you can make a change that considered a minor change that do not have any additional requirement other than demonstrating quality aspect, which is meeting the new product made by the batch size change shows the same QC testing profile than the pre-change.
Well, thank you for answering, and thank you, audience members, for your questions. Before we finish the webinar, we'd like to ask our presenter once more, that's Dr. Xin Bu, if they have any closing remarks.
No. Thank you. I really appreciate this opportunity and hope the information provided is useful information for the audience.
Well, thank you. I would like to take this opportunity to thank our presenter for sharing their knowledge with us. I'd also like to take this opportunity to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you are watching this on demand, then please feel free to send your questions over to me at stephen.edwards@biopharma-asia.com, and we'll get those answers back to you. Thank you, everyone. Have a great day.